Here we are, biology students. We've arrived to our final unit, and this unit is 5 chapters long, and it's all about genetics. So chapter 10.1, sexual reproduction and genetics. Section 1 is about meiosis. Section 2 is Mendelian genetics. Section 3 is gene linkage and polyploidy. The big idea for the entire chapter of sexual reproduction and genetics is reproductive cells, which pass on genetic traits from the parents to the child, are produced by the process of meiosis. So section 1 is going to be all about meiosis produces haploid gametes. Section 2, Mendel explained how a dominant allele can mask the presence of a recessive allele. And section 3, the crossing over of linked genes is a source of genetic variation. Now at this point you're going, I don't know what a haploid is. A gamete what? Allele crossing over, like Red Rover, send Mrs. Lewis right over? I don't get it. Well, if you don't, that should tell you there are going to be a ton of new words for you to learn. So make sure you learn them. If you remember in the last segment, the video talked about mitosis and meiosis are two totally different things. And you have this available to you on your online textbook as well as in your textbook. I would encourage you at this point to make a graphic organizer yourself. Just copy the one right out of the book and study that so that you understand the differences. Although we're done talking about mitosis for the most part, it's still going to be relevant for the rest of this unit. So meiosis produces haploid gametes. Well, what does all that mean? So it's all about chromosomes and the chromosome number. Characteristics such as hair color, eye color, all those types of things, the size hands you have, these are all called traits. The instructions for each trait are located on chromosomes in the nucleus of the cell. Remember your cell city. DNA, deoxyribonucleic acid, organized in segments called genes that control the production of a protein. Each chromosome contains hundreds of genes, and I would like to remind you at this point, you will want to use your colors. Yes, you have to color. Now, homologous chromosomes, homo, the same. That's what it means. It's a scientific word. Human cells have 46 chromosomes or 23 pairs. One contributed by each parent. That means you get 23 from mom and 23 from dad. The chromosomes that make up the pairs are called homologous chromosomes. That means they're the same length, the same centromere position, and carry genes for the exact same traits. Haploid and diploid cells. To maintain the same number of chromosomes from generation to generation, organisms produce gametes. What are those? Well, that's the fancy word for sex cells with half the number of chromosomes. The symbol N can be used to represent the number of chromosomes in a gamete. So a cell with N chromosomes is called a haploid cell, and a cell that contains 2 N chromosomes is called a diploid cell or a diploid cell. So here we are, meiosis 1. Meiosis is a type of cell division that reduces the number of chromosomes in a cell and produces gametes, and it involves 2 consecutive cell divisions, meiosis 1 and meiosis 2. So here we have a female, and she's undergoing meiosis, and she produces a female gamete. It's a haploid. It's an N. The male undergoes meiosis, and he produces a gamete known as a sperm. So we have an egg and a sperm. Both the egg and the sperm contain 23 chromosomes each. They come together, they're fertilized, and then all of a sudden we have a diploid. A diploid is a zygote. It's a baby in a cell form before it starts growing. And now we have 46 chromosomes. During the growth stage, turns into baby boy or baby girl. So meiosis 1, interphase. Chromosomes replicate and chromatin condenses, just like before. Prophase 1, pairing of homologous chromosome occurs, and each chromosome consists of 2 sister chromatids, kind of like the same as before a little bit, right? So as homologous chromosomes condense, they're bound together in a process called synapses, which allows for crossing over. Synapses and crossing over are 2 concepts that you will want to most definitely pay attention to. Crossing over is when chromosomal segments are exchanged between a pair of homologous chromosomes. Crossing over produces exchange of genetic information. So here you can see we have a set, and then all of a sudden it's changed. He's taken a little bit of this and they've just swapped them. Okay, here again, they've swapped. So metaphase 1, chromosome centromeres attach to spindle fibers, while homologous chromosomes line up as a pair at the equator. During anaphase, stage 1, homologous chromosomes separate and move to opposite poles of the cell. The chromosome number is reduced from 2N to N when the homologous chromosomes separate. And you should know why. Make sure you're paying attention. There's a reason why it has to reduce. Telophase, chromosomes reach the cell's opposite poles and cytokinesis occurs. So now we're done with meiosis 1. Now we're on to the second cycle of meiosis, and it's prophase 2. So a second set of phases begins as the spindle apparatus forms and the chromosomes begin to condense. Metaphase 2, chromosomes are positioned at the equator, and meiosis 2 involves a haploid number of chromosomes. That means they have half of what they had originally. And finally, we have cytokinesis, which results in 4 haploid cells, each with an N number of chromosomes. Now I'm going to have more of a visual on the importance of meiosis and how that happens in a later video. But it's important because mitosis consists of only 1 cell division that produces identical cells. Well, wouldn't the world be boring if we all looked the same? So meiosis consists of 2 cell divisions instead of 1 that produce haploid daughter cells, and those cells are not genetically identical. There's actually 4 different variations. So, in a nutshell, meiosis results in genetic variation. Now, it's important to point out when we're talking about variation that during prophase 1, the chromosomes line up randomly at the equator, and gametes end up with different combinations and chromosomes. Genetic variation also is produced during crossing over and during fertilization when gametes randomly combined. This should be gametes. Now, while we're on this topic, it's important to understand there's 2 types of reproduction, sexual and asexual. Asexual reproduction, the organism inherits all of its chromosomes from a single parent. The new individual is genetically identical to its parent. And sexual reproduction, the rate of beneficial mutations is faster. Beneficial genes multiply fast over times than they do for asexual organisms because of genetic variation.
Have you ever wondered how 2 siblings who could have the same mom and dad can still look so different? Well, today we're going to talk about the process that makes that possible, a process called meiosis. You might remember that humans have 46 chromosomes. That's how many all of the body cells in your body have. But there's some cells in your body that don't have 46 chromosomes. Human sperm cells and human egg cells actually only have 23 chromosomes. Why? Well, think about it. If a human sperm cell has 23 chromosomes and a human egg cell has 23 chromosomes, when they come together, that makes 46. That will now allow a new developing fertilized egg to develop into a human. Meiosis is all about sperm and egg cells, or, if you want to get really fancy, gametes. Gametes is definitely the fancier word. Meiosis is what we call a reduction division, because you have a starting cell that has 46 chromosomes in humans, and your ending cells only have 23 chromosomes. So it's reduced. It's a reduction division. And in its production of sperm and egg cells, the cells are going to be all different from each other. In males, remember males make sperm, all the sperm cells are going to be different when they're produced. They're not identical cloned sperm cells. This whole process is really different from mitosis. Mitosis was really great when it was going to repair damage or worn out cells. Mitosis was really great for growth because mitosis makes body cells. Mitosis has to make identical cells. Like when you have a cut and you have these damaged skin cells, you have to replace them with identical skin cells. That's what mitosis was all about. Meiosis is not about making identical cells. The cells that meiosis makes are not identical, and they're going to be sperm and egg cells. Now, before we get into these stages of meiosis, we have to remember what comes before meiosis. This actually also happened in mitosis. It's that stage known as interphase. Interphase, it's when the cell's growing, it's replicating its DNA, it's carrying out cell processes. Just like mitosis, interphase happens one time. So it happens before meiosis is going to start. And you might remember from mitosis that little phrase PMAT. The P was for prophase, the M was for metaphase, the A was for anaphase, and the T was for telophase. And the good news is, in meiosis, you will still use those terms. So that's great. But because meiosis is actually a reduction division, you're going to divide twice. You're going to do PMAT twice. So we're going to go through it so that it's going to make more sense. Here's the good news, though, about PMAT happening twice. In meiosis, the stages have numbers after them. So there is no prophase in meiosis. There's a prophase 1, and there's a prophase 2. And there's no metaphase in meiosis. There's a metaphase 1 and a metaphase 2. So step, prophase 1. One thing I like to remember about prophase is the word pro. If you're talking about the Greek root, it actually means before. So it kind of helps to remember that it comes before all the other stages. This is where the chromosomes are going to condense and thicken. They're actually going to line up with their homologous pairs. The word homologous means that the chromosomes, they're approximately the same size and they contain the same types of genes in the same locations. So they're going to match up. And it's during prophase 1 that this amazing process occurs. It's called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really neat process because when these chromosomes are lined up in homologous pairs, they have this way that they can transfer their genetic information and exchange it between each other. It makes for recombinant chromosomes, which can contribute to the variety that we mentioned, that siblings, even if they have the same parents, they can be different. Well, this is a process that contributes. Next, we move into metaphase 1. I always like to remember the M for middle. In metaphase 1, the chromosomes are now going to line up in the middle. It's a little different than how it is in mitosis, though, because they're still in pairs. They're homologous pairs, so they're not going to be in a single file line. Next, anaphase 1, think A for away or for apart, because this time the chromosomes are going to move apart. And so this time you can think of the T for 2, at least for now, at the end of meiosis 1. Eventful of having the process of crossing over. In metaphase 2, again, remember M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. Even though it's actually the chromatids that are going to be pulled apart, chromatids are getting pulled apart to opposite sides of the cell. And in telophase 2, remember, we had 2 cells that we're going to divide. So now, if they both divide, we are going to end with 4 cells. Now, scientists are often looking into the process of meiosis, because sometimes those chromosomes, they don't separate correctly. It's called non-disjunction, when maybe a cell receives too many or too few chromosomes because they do not separate correctly. This can contribute to some genetic disorders, which is something scientists are often studying, and when we get to genetic disorders, we'll talk about that relationship with meiosis.